want to welcome all of you. It's really fantastic. We have an amazing lineup of speakers. We just want to start by thanking our affiliates. Ben mentioned already, Papam Biotech, Nova Nordis, PNC, CHI, and Trevo are all sponsors of today's and other events that we have running. So thank you very much to them. So just for fun, I thought I would say, ask ChatGPT, what are the hottest areas in the biological sciences? And they came up with these 7 categories. I didn't try it again, so I don't know if you get the exact same 7 again, but you probably would. And what I thought was kind of interesting is that when I looked at this list, at least 5 of these, maybe all 7, I'm not sure. We'll see what the speakers have to say. But at least 5 of these are going to show up in the talks today. So we're covering 5 of the 7 hottest areas of biology, and some of them you'll recognize just from the speakers that are coming. So here they are. They're going to be covering these various topics that really will span a wide range of very exciting areas, mostly centered around RNA, but all kinds of biotechnology and other areas as well. Artificial intelligence, I'm sure you appreciate, is infiltrating all areas of healthcare and biology. We think about this a lot at Stanford, but these are just some of the areas which AI has embedded itself into healthcare in general. So patient management, diagnostics, treatment selection, outcome monitoring, designing therapeutics, and certainly relevant to today's symposium, research is clearly a huge area in which AI is becoming very, very prominent. And just for fun, I thought I'd give you a few examples of some of the areas in which AI has touched itself in our research. We think it's really transformed a lot of what we've done. And just for fun, I'm going to show you some of these examples. One is the genetic basis of human disease. You probably know most human diseases are complex, meaning they're thought to be polygenic in the sense that lots of common variants are thought to affect whether you're at risk or even protected for a common disease. And so what people do when they try and make these predictions about risk, they do something called a polygenic risk score. They'll sum, you probably know, we have lots of common variants that are different between us. And so what people do polygenic risk scores do is they'll sum over literally these days thousands, if not millions of variants to try and make predictions about people's risk. And to be honest, it doesn't work very well. And it doesn't work, it may work for the top 2 or 3%, but it really doesn't go that deep. And I think the reason it doesn't work well is twofold. One is these are generally linear additions and of sort of GWAS and other signals. And of course, genetics isn't really linear, so it doesn't make sense. It should work well. And by that, I mean, if you have 2 mutations, same pathway, 1 + 1 is 1, 2 mutations, different pathways, 1 + 1 can be 15. So linear addition isn't going to cut it. And not only that, they're missing rare variants, which are highly penetrant. That's left out of these analyses, is some of these common variants that are thought to be a low effect. And so one way in which we've been using AI is to try and actually analyze complex disease. And I'll just show you we're using 2 approaches for this. One is rare variants, and the other is these common variants. And so starting with the rare variant one, I'll show you by example. There's a known condition, abdominal aortic aneurysm. It's semi-common. 10% of people over 65 die of this. And it's discovered when their aorta bursts. So it's a terrible way to know you're at risk. And then they, of course, die usually when that happens. It's known to be highly heritable, 70%, but only 3 sort of questionable genes have been identified when we started our study. So what we did was we actually went in and sequenced 268 cases, 133 controls. Now that'll sound ridiculously low to someone who does genome-wide association studies, but nonetheless, that's what we did. And then we called pathogenic variants. We looked inside at the exons and called known as well as likely pathogenic variants. And then doing machine learning with the 10-fold cross-validation, we added in a little EHR, and we actually came up with 60 genes, incredible p-value, 10 to the -13. And we're pretty convinced these are right because they're involved in the pathways you would expect. Mouse models of these 60 genes have A defects, and the genes are misexpressed both in mouse models as well as in AA patient. So using machine learning, we could just from a relatively modest number of genomes really get at a pretty a deep, set of genes that are involved in a pretty important disease. And we could even go on and build polygenic, if you will, polygenic risk score from the genes we identified. It's not too bad. The genome alone is about 0.7. Then you add a little EHR, you get 0.8. It's as decent as any, say, Framingham thing. So the point out of all this is, though, that we actually could get at the genetic basis of this much better. We've gone on actually and gotten at some other genes that are also known to be highly heritable, other conditions. ALS is about 60% heritability, and there were 7 known genes. We use a different approach here. I won't have time to get into it, but you could do, we can map out open chromatin regions in motor neurons where ALS is thought to occur. And then we can actually we ask for which ones have an enrichment for GWAS signals. GWAS in general, many of you know, is thought to have its signals in regulatory sequences which lie in open chromatin. And the amazing thing was, from a fairly large GWAS study of about 12,000 patients, 24,000 controls, we went from 7 genes that have been discovered with that exact data set to 690 genes. And this is turning out to be common. We'll find anywhere from 10 to 100 times more genes using these new AI ML methods. And feel free to look this up. I know I don't have time to do this. And we went on even further in severe COVID, which is when people are young, they get severe COVID. That's thought to be a genetic effect as well. And there we went from 47 known genes over 1,000. And we could even identify the cell type with the approach we use. Turns out natural killer and T cells have most of the genetic signal. And in each case, we came up with, this case, 36% of the heritability, this case, 77%, which is pretty amazing. So these new AI methods are turning out to be very, very powerful for genetic basis disease. Many of you know we also do a lot of work with wearables, and we can tell, as Ben mentioned, when somebody's getting ill, in fact, even COVID, in advance of symptoms with a smartwatch. I'm not going to show you that today, but we've gone on to write machine learning algorithms for detecting COVID, but it turns out you can detect quite a bit of information just from simple features in a smartwatch. Again, using AI and machine learning. Here we use Lasso. And what we did is to determine whether we could grab features from a smartwatch. Smartwatch measures many, many things you may know: heart rate, resting heart rate variability, SpO2, etc. This is a very old smartwatch. And even with this old smartwatch, we could get, these are clinical signals: hematocrit, red blood cell count, hemoglobin. They're involved in anemia. And we can actually get a decent signal just from features on a smartwatch using machine learning. Same with hemoglobin, A1C, fasting glucose. So we think these methods, again, are very, very powerful. We've used them in other ways. These are conflicted here. My company, January Eye, they actually will measure from 4 days of continuous glucose monitoring. They can actually make very, very accurate predictions of your glucose levels and actually use a whole AI program to better manage your glucose. And these days, they can even predict your glucose without a CGM, continuous glucose monitor. It's pretty amazing. So feel free to visit them out. And then lastly, another way in which you can use AI is also in returning information. And that's what one of my other companies does. Again, conflicted here, but this is a microsampling company. They basically collect blood, and you mail it in. They'll measure over 500 analytes that are they span over 20 wellness areas, and they'll give you a report back on those areas, as well as even your aging and your aging subtypes, if you will. We call them ageotypes. And the bottom line is that with AI, you can actually pull in information and actually make lifestyle recommendations based on the data that's pulled in. So the bottom line is that medicine and science these days is very information rich. And from that information, it's really quite easy to start using these modern tools to better analyze your data using, various methods, including deep learning and such. And so the bottom line is AI is everywhere. If you can turn your data into features, you can do AI. Now, you do want to have enough data so you don't get biases, and you also would like to have representative data as well. And so you can also, get new results, like I mentioned, genetic basis of disease. but you can also use AI to actually collect other information that's out there, pull it in, and make return of information and conclusions from all these data. So that's what I wanted to say. AI is everywhere. I'm going to be curious to see how it permeates the various talks we have today. And we have an amazing lineup, as you've heard already. And so this is the list. So please stay with us for the entire day as much as you can do. And that's what I wanted to say. I guess I do have a minute or two if anyone has some questions, but otherwise I can give more time to the first speaker if she's online yet. So does anyone have any questions about any of that? Again, I think AI is coming. I see Jennifer is here. Fantastic. All right. Why don't we just jump in there? As I say, my main goal is to welcome all of you. And I am going to have the pleasure of introducing Jennifer Doudna. And I've actually known Jennifer for a long, long time. We go back to Yale days together. I think we're both assistant new professors trying to figure out our way in the world, the academic world. She moved to Berkeley a number of years ago. I've lost track of what time. I guess in a sense, I followed her when I moved to Stanford several years later. Anyway, she doesn't really need any scientific introductions. Everybody knows her. She's done fantastic work. But I think what's special about Jennifer, she's not only one of the most amazing scientists I know, but she's also, you know, a pretty amazing person who's always been very generous with her time. And she'll probably get bombarded with questions by the end of this thing, but I know she'll make herself available as much as she can. And of course, you want to know Nobel Prize and all that kind of stuff too. So that's nice. So anyway, Jennifer, I'm able to give you 3 more minutes for your talk if you would like to use them. And so welcome, great to have you here, and I'll turn it over to you.